Okay, welcome everybody. Morning to the third day. My name is Joachim Schulze. I'm from Bonn in Germany, and I have the pleasure to chair the session, the first session. Christoph, there is no extra opening, right? You have no, so we start immediately. People move in. We have 3 exciting speakers. We're going into the medical impact session. And I'm delighted to ask Alexander Marson to the podium. And he's telling us about his efforts on decoding and reprogramming human immune cells with CRISPR. Alex.
Good morning. Well, I just want to thank the organizers and everyone for welcoming me into this community. It's been amazing and inspiring to hear about the technologies, the data, the analytic tools, and the insights that are starting to emerge from this group. So my goal this morning, I think we've heard a lot about the opportunities for the Cell Atlas to inform us about the composition of the human body, how cells are altered in disease states. Today I'm going to argue that there's an additional opportunity for the Cell Atlas to not only inform our understanding of disease pathology, but actually to inform our understanding of disease treatment. And where I think there's a special opportunity is in the emerging field of genetically engineered cell therapies. And so many people are now inspired by the successes of genetically engineered immune cell therapies. And I think we're just seeing the tip of the iceberg of what's possible. So this is the farthest along in the treatment of cancer in the form of chimeric antigen receptor or CAR T cells. And the basic idea of this is that patient cells are taken out of a blood sample. They're genetically engineered outside the body. In the case of CAR T cells, that's by putting in a transgene with an artificial receptor that redirects the specificity of T cells against a cancer antigen. And then these cells can be reinfused back into patients. But in theory, the opportunities are limitless about the genetic edits that we can put into cells. And this is not necessarily limited just to cancer. Although I'm going to focus on cancer today, there's huge opportunities in autoimmune disease, infectious disease, and the basic idea is that immune cells are already in circulation. They have evolved to treat disease, but we now have new tools to actually reprogram them to enhance their ability and really direct their specificity and program in specific functions that will make them into better therapies. And of course, this is supercharged in an era where we now have genetic tools of unprecedented power, right? So CRISPR often gets the most attention for the ability to fix individual mutations that are causing rare monogenic diseases. But I think there's even more possibilities in the era of synthetic biology to imagine how CRISPR can actually go in and target specific gene programs to make cells behave how we want. So we can remove genes that are limiting the function of cells, or we can actually take whole stretches of targeted locations in the genome and add new sequences with synthetic biology codes to replace the existing code to really program in exactly phenotypes that will make cells into effective cellular medicines for disease. So I will focus on the problem of cancer initially, but I think the applications are even broader. So in the field of cancer, I mentioned that CAR T cells have been really miraculous for certain forms of cancer. For certain types of leukemia and lymphoma, incurable cancers have now been cured, but the majority of cancers still don't respond. And that problem is particularly severe for solid tumors. And we know that there are a number of challenges in the solid tumor field, but among them, even if we can make an antigen-specific cell, an antigen-specific T cell that will recognize a solid tumor target, either by putting in a CAR or a defined T cell receptor, these cell therapies face a number of challenges once they get into the tumor microenvironment. So they'll have to be chronically stimulated, which can cause dysfunction. There's checkpoints, there's suppressive factors in the microenvironment, there's suppressive cells. So we'd really like to design a cell therapy that not only has the right specificity, but also has enhanced function to overcome all of these challenges. And the way that we've been thinking about it is that we'd like to be able to define an ideal profile of what a cell therapy would look like. We'd like it to, once it recognizes its target, continue to proliferate, resist all these modes of suppression that I told you about. We'd like it to be durable over time. Cancer cells can turn down their targets. We'd like these T-cell therapies to recognize even low antigen density cancer cells. And through all this, we'd like them to be effective at clearing tumors. So once we have a target profile in mind, we have really turned to the power of forward genetics to think about how can we figure out what are the genetic edits that we can put into a T cell to endow them with these properties that would make them into a powerful medicine. And the idea of forward genetics, as you know, is basically to make a lot of perturbations in a population and then pull out the individuals that have a phenotype of interest. And in this case, the population is a population of primary human T cells, and the phenotypes are the ones I just told you about, the cellular properties that are therapeutic. So we have defined and adapted and developed a number of tools for high throughput perturbations in primary human T cells. And I'm just going to very briefly today introduce them. We can now do genome-wide knockout screens. And more recently, and I'll show you today, we can now not only do knockout screens, but we can do gain of functions with Crispr activation. And I'm not going to have time today, but I'll just briefly end by saying, beyond turning genes on and off, we've also developed a technology where we can pick one site in the genome and knock in a whole library of different synthetic DNA sequences into a defined site and make a population with different knock-in sequences. And what all these technologies have in common is that you end up with a pool of cells that have been perturbed in different ways and are effectively barcoded so that you can track which perturbation has been introduced in each cell in the population. Once we have this pooled cell with all these perturbations, we can race them against each other in a number of different ways. We can just see which ones are the most fit, which ones will increase in abundance over time, especially in different challenging contexts, in vitro or in vivo. We can also use FACS sorting or other selections to pick out cells that have defined phenotypes of interest based on markers that have been well identified. Or where I think there's huge opportunities is combining these with single-cell profiling technologies, including single-cell RNA sequencing, to really match up each perturbation with its overall effect on cell state. And this is where, over the past couple of days, I've just been filled with ideas of how we can use various emerging technologies to even more rapidly identify the perturbations that move the cells to phenotypes of interest. So I'm just going to take you through these three different types of perturbations and how they're informing design of cellular therapies. So there's now been many, many genome-wide CRISPR screens in cell lines and in animal models. We, a number of years ago, faced the challenge of how do we take this that was so successful in cell lines and bring it into primary cells. And initially, we had a lot of trouble getting the Cas9 machinery stably transduced into T cells. So two postdocs, Eric Schiffrud and Julia Carnevale, who now have their own labs, they came up with a clever strategy to do a hybrid technology where we can put in a library of guide RNAs with lentivirus, and then we electroporate Cas9 protein. And the Cas9 is able to find the guide RNAs expressed off these lentiviral cassettes, and it actually leaves a permanent genetic modification. So we can then race these cells against each other in any way, and they've adapted to this to genome-wide scale. This was published a few years ago, but where we've taken this, where Eric and Julia have taken this, is they've now done genome-wide screens in a number of different in vitro conditions that reproduce different aspects of that suppressive microenvironment that I told you about that challenge these T cells. And all of these many of these genome-wide screens converged on a new gene that has never been studied in T cells before, called RASA2. RASA2 acts really as a checkpoint that limits the function of chronically stimulated cells. And what Eric and Julia have found is that if they remove RASA2 from CAR T cells, the cells are not only more sensitive to low antigen, they can resist immunosuppressive features in vitro, and most importantly, in preclinical models, they outperform control cells. So this is with CD19 CARs knocked into the TCR locus. And for those of you who know, these are probably the best CAR T cells that are out there. And we have found that by knocking out RASA2, we can make these cells even more powerful. So they can perform at lower stress, at doses and where the control cells are not able to control tumor volumes, the ROS2 knockout cells control tumor volumes and prolong survival. So I think this is an example of how we go from genome-wide screens to candidate identification all the way to preclinical models. And I should say our colleagues at St. Jude's have actually also tested this in a solid tumor model of sarcoma, and it's also ROS2 knockout there is also improving outcomes in preclinical models. So we think we've really found a new module that we can modify to improve cell therapies fairly widely. But of course, loss of function is only a piece of the puzzle. We'd also like to not only be able to ablate every gene in the genome, we'd like to be able to turn every gene in the genome on and see what effects those have on cell therapy. And the tools for that have also been well established in cell lines. And in particular, there's Crispr Activation, where CRISPR is transformed into an artificial transcriptional activator by fusing a nuclease-dead version to the VP64 and other transcriptional activators. There's also a CRISPR interference, which is a transcriptional repressor. We couldn't use these tools in primary cells because of their size. We weren't able to stably transduce them into human T cells. And we tried for many years, but basically 2 postdocs have systematically tackled this. They've improved the transduction efficiency, Ralph Schmidt and Zach Steinhardt, and very quickly they were able to go to get efficiencies that now are compatible with genome-wide CRISPR activation screens in primary human T cells. So they can use existing genome-wide libraries compatible with CRISPR-A, put them into primary human T cells, and again, now you end up with a pool where a set of human T cells where a different gene has been turned on in each cell. And we can race those cells against each other for any phenotype we're interested in. What Zach and Ralph have started with is to learn the rules of every gene that controls cytokine production. So cytokines are the signaling molecules that are critical for T cell function. We'd like to be able to control that for cell therapies. So what they did was actually FACS-sorted cells, either the CD4 cells or the CD8 cells in a population, and they sorted for intracellular staining based on different levels of production of either IL-2 or interferon gamma, 2 cytokines in these cells. So these are not reporters, these are endogenous cytokine production inside the cell. They're able to actually do these big FACS sorts and learn the rules of which genes control production of these cytokines. So this data immediately grabbed my attention. This is exactly the kind of data that you hope for. You can see that what I'm showing you here for the IL-2 and the interferon gamma screens are replicates from 2 human blood donors. And you can see the replicates line up incredibly beautifully on these diagonals, suggesting these are really robust genetic screens, reproducible in different human blood donors. And the hits make sense. So if you directly transactivate IL-2, that comes up in the high IL-2 bin. If you directly transactivate interferon gamma, that's the top hit for interferon gamma. And it's not only reproducible at the level of genes, it's also reproducible at the level of guide RNAs. So you can see multiple different guide RNAs that are in the expected bins for IL-2 and interferon gamma, and for other genes that are known to be either positive or negative regulators of T cell responses. So we've really now put together a map of genes that we can turn on to start tuning cytokine production. And what turned out to be particularly informative is that Zach and Ralph repeated these screens, not only for CRISPR activation, but for CRISPR interference for the same phenotypes. And that turned out to be interesting. Maybe this should have been obvious in retrospect, but it was surprising to me and really caught my attention. If you look at the expression levels in T cells of the CRISPR-I hits versus the CRISPR-A hits, they're quite different. The CRISPR-I hits are already stably expressed at significant levels in T cells, which makes sense. They have to be on in order to have a phenotype when you turn them off. But the CRISPR-A actually are expressed across a wider range of expression levels, suggesting that genes that are off can actually be turned on to give you a phenotype. And that can either be because these are genes that are only context restricted and get turned on in certain contexts, or it could be that we're really finding synthetic functions and repurposing genes that might be normally used in a different cell type, actually repurposing them to regulate T cell functions. So I'll give you one example of this. This is comparing CRISPR-I to CRISPR-A. The CRISPR-I hits are on the x-axis, the CRISPR-A hits are on the y-axis. And we labeled for regulators of interferon gamma the hits that fall into the NF-kappa B pathway. And you can see that in the NF-kappa B pathway, there's some hits that come up in both screens, but some only come up in CRISPR-I and some only come up in CRISPR-A. And when we look at what these genes encode, it actually starts to make sense. CRISPR-I is very good at picking out the intracellular machinery, and we think that that's because each of these genes is individually required, they're already expressed at baseline. In contrast, CRISPR-A picks up a whole bunch of cell surface receptors that were missed with CRISPR-I. So we think that these are not individually required, but they're sufficient to regulate interferon gamma when they're turned on. And these are not just a random set of genes, they're actually, if you just by visual inspection, they include many genes that are already under active development for immunotherapies, either with drug development or for genetic modification in cell therapies. And that includes OX40, 41BB, CD27, CD40. So we think we found a new treasure trove using this gain-of-function screen technology. And I'll just now briefly say that these tools are compatible with Perturb-seq approaches developed by Aviv and others, where we can actually couple these perturbations with single-cell readouts. So we were able to find a way to recover the guide RNAs that are introduced with CRISPR-A in a 10x platform, and very readily we were able to test 70 different genes, including controls, but many genes that have not been well characterized from our screens, either positive or negative regulators of the cytokine production. We put these into cells from 2 human blood donors, and what I'm just showing you here is a dimension reductionality map showing that the 2 donors, and we were able to look at them based on genetic SNPs. You can see there's not a lot of donor separation between these cells in cell space, which is good, but we are able to distinguish between the CD4 and the CD8 populations in this population of cells. And then we can look in and we can see at the genes that we initially screened for. You can see there's populations that produce interferon gamma. IL-2 was a little less well captured in the single cell, but you can see there's a CD4 population that's producing IL-2. And we were able to sort of come up with an overall gene score for T-cell activation. So this was just comparing differentially expressed genes between resting and activated cells and looking at, across this space, the extent to which these cells appear to be activated. And then we can look and see at what the effect of each of the perturbations are. So as you expect, the non-targeting controls are relatively evenly distributed, suggesting they're not having any effect. But if we look at the perturbed cells, we see that they start to occupy different spaces on this map, including some new spaces that are not in the non-perturbed cells. And then we can look at individual perturbations, and you can see, for example, MAP4K1 is keeping these cells in a state of low activation, consistent with its low cytokine production. And specific guides, like GATA3, move the cells to a Th2 state. TBX21 moves the cells to Th1 interferon gamma producing states, or CD8's the interferon. So you can actually see multiple states promoted by TBET, and then another example of cells that move the cells to another interferon gamma producing state. So I'll just briefly, this is the last slide of data, to show that we can actually cluster in a semi-supervised way all the cells in this space. And then we start to annotate, as we've discussed a lot at this meeting, the different states in this map, and we can actually look at each CRISPR-A perturbation to see which overall T-cell state has been promoted, either a natural or a synthetic state that has been produced by overexpressing this gene of interest. So I think we're really entering into an era where we have the lexicon to start thinking about loss of function and gain of function genes that can be used as the building blocks of synthetic codes to make cells move to specific states or to turn on or turn off gene programs that modulate their therapeutic function. And as we have these building blocks, where I think this is heading, CRISPR-A will not be the ultimate therapeutic modality. You can't keep CRISPR-A in T cells, it'll be immunogenic if you're going to put them into the body. But what it will tell us is how we can start to write DNA sequences that can then be knocked into specific sites in the genome with CRISPR. And so this is where we're using pooled knock-in technology to write combinatorial codes into multi-cistronic DNA that encode both an antigen receptor and some other gene of interest. And we're coming up with many hypotheses based on these screens and from other sources where we can knock these different synthetic codes into T cells. And then, once we have this population, we can race them against each other in vitro, in vivo, or even recover single-cell codes with single-cell RNA sequencing. So I'll just end by saying I think that now what I've tried to argue is we have the tools to actually go in and understand the cell circuits that control human immune cells. And that's going to tell us fundamental lessons about how these cells are wired. It's going to give us insights into what's failing in disease states. But now each of these tools, we can actually start to perturb coding and non-coding sequences to learn the rules of synthetic programs that we can put into human immune cells to make them more effective for treating cancer or autoimmunity, infectious disease, and I think other classes of human disease as we get more advanced in synthetic programs to move new functions into these cells. So with that, I want to thank the people who've done this work and our funding sources, and thank you again for having me.
Yeah, Alex, thank you very much. Very exciting new technology. There's a question over there.
Hi, great talk, thanks. So I wonder whether you can comment on efforts to leverage the wealth of diversity, cellular diversity that is known from the HCA to engineer T cells or CAR T cells beyond sort of their targeting capability, but let's say to make them go into a specific organ or a specific niche, and therefore repurpose them for applications beyond oncology.
I think that's a fantastic question. So I think the opportunities are really clear. That I talked about the limitations from the tumor microenvironment. Another major limitation for T cell therapies is finding the right targets. And the level of single cell data that's emerging from the HCA is a treasure trove of thinking about what are the targets that are the most selective, either alone or there's now combinatorial codes for antigen specificity that can be used to find combinations of specific targets that will really direct T cells to a specific site. And we really need to know that it's specific and that there isn't the risk of an off-target. And if we can make T cells go to a specific site, then we can use synthetic biology to not only make them find that site, but respond and potentially secrete payloads. So I think we can also learn lessons about what payloads might be therapeutic and promote wound healing or treatment of other types of pathologies. So I think that this ability to code antigen specificity and function is greatly informed by the efforts of the Human Cell Atlas.
We have two more questions. First, Aviv, and then Jose.
Hey, thanks, Alex. It was a great talk and beautiful work. And couldn't agree more with your previous answer. Absolutely. That's why we need an atlas. But I have a question on the Crispr Genome-Wide versus the Perturb-seq that followed.
Yeah.
So in the Perturb-seq, you showed that actually the different perturbations led to very different cell states.
Yep.
Are they still all perturbations of genes that in the genome-wide screen basically had the same phenotype? And was that phenotype also observed in what you would see in the single-cell profile?
Yeah, so more or less yes is the answer. So the hits were selected based on levels of IL-2 or interferon gamma, and generally the things that were associated with high levels were again recapitulated that. Again, IL-2 was less well captured in the
RNA.
In the RNA-seq, so it's not perfect, but overall the effects tended to be highly consistent, yeah.
Right, thanks.
Last question.
Yeah, Alex, very exciting work. I had a general question for you. You showed two nice examples where you had a high-plex genome-wide screen and then a focused set of singular cytokine readouts, and then maybe a reduced amount of different targets with a higher plex readout.
Yeah.
Which are you most excited about in terms of carrying forward, in terms of where you've learned the most maybe exciting or unexpected biology?
It's a good question, and I think it's a little hard for me to answer it. We've learned the most from the scale, right? To be able to explore the unexplored. Like, for example, finding ROSA2 is not a gene that we would have put into any Perturb-seq to start. So in some ways, the best solution would be able to do genome-wide with deeper phenotyping. I think in some ways, we're still in these early stages where being able to do things at scale to pick out novel hits is probably the most informative, but as things move forward, we'd really like to be able to understand the full effect on cell state and function.
Alex, thank you very much again. We move on. Thanks. Give him an applause. And we continue with Mark McCarthy from Genentech, and he will tell us about human genetics meets single-cell genomics. Mark, all yours.
Hi, good morning, everyone. Thanks for the invitation to be here. It's the first time I've come to a Human Cell Atlas meeting. It's been a revelation. See how far the science has come, and hardly seem to have skipped a beat despite the last 2 years of interrupted science. So yeah, I'm going to give you a little bit of a flavor of some of the things we're thinking about at Genentech in relation to single-cell genomics and single-cell atlases and the like, particularly from a human genetics perspective, because I think there are huge opportunities that come from, in the coming years, integrating the power of human genetics with the insights that come from single-cell genomics. So very high level, one of the things that's a real focus for us at Genentech is to take advantage of what Aviv calls these multiplicative levers, these new approaches, these new technologies that we think can help tilt the balance in favor of a more active and successful approach to drug development. And 3 of those at least: the focus on human biology, the use of techniques that access high-resolution genetic and genomic data at scale, and the advanced computation and machine learning that's needed to make sense and interpret those data really overlap with the mission of the Human Cell Atlas. So no big surprise that single-cell genomics and use of human cell atlases is an important part of what we're trying to do. And we use these techniques, these high-throughput genetic and genomic techniques, to understand the processes that are involved in disease onset and progression, because those are pointers to new therapeutic approaches that we can seek to exploit. But actually, a lot of the time, our problem isn't that we don't have targets, it's that we don't have good targets, by which I mean targets where we can feel confident that we can turn those into safe and effective medicines. So we need to use the same approaches to think not just about target discovery and identification, but also how to prioritize those targets and how to progress them efficiently through the drug development pipeline. And that involves actually thinking beyond relationship to causation of disease onset and progression, but to tackle other questions to do with prediction of safety, biomarker identification, and tractability, and so on, which are also obviously very important components if we want to turn those insights into new therapies. Now, you wouldn't expect me to say otherwise, as somebody who comes from the human genetics field, but human genetics is an important part of all of this. It is, after all, in vivo functional biology in the species that we care most about. It provides us with equivalence in terms of perturbation between genetic perturbations that happen in natural experiments that we detect with human genetics and the clinical experiments of therapeutic perturbation that we seek to achieve. And it also provides us with strong causal inference. There's no surprise that, as a result, targets that have got human genetic support have roughly twofold the probability of therapeutic success of targets that do not. And as I said earlier, really part of the challenge over the next few years is to learn how to bring that power that comes from human genetics, particularly as it relates to causal inference, together with some of the single-cell genomics, particularly as we start to think about single-cell changes that we might detect in disease versus normal tissue, where it can be pretty hard to understand what the causal relationship between those changes are. So we can use this is pretty simplistic, but we can use the causal inference that comes from human genetics and ally it to the mechanistic inference that comes from single-cell genomics, then we'll certainly be in a far better place. We need to deal with the fact that we're dealing on different scales often. Obviously, with human genetics, we're often dealing with sample sizes now in the millions. It's great to see some expansion in the single-cell space to start to consider increased sample size to capture genetic variation and to capture ancestral diversity. So I thought I'd just take you very quickly through 3 approaches that we're using to combine those 2 worlds. Now, the first, the most obvious, is one borrowed from work that's been done with bulk expression data, for example, which is to overlay genetic signals onto single-cell derived annotations. That works very well for bulk data, be that RNA-seq or ATAC-seq, and of course it works incredibly well for single-cell data. Here's an example of a human cell atlas from retina generated by colleagues at Genentech, overlaid with Genome-Wide association data for age-related macular degeneration, pointing to the likely importance of retinal pigment epithelium cells in terms of the expression of many of the genes that are emerging from those genetic studies. And here's another example from Alzheimer's disease. You're probably familiar with the idea that if you overlay Alzheimer's disease GWAS onto those cell type specific annotations, you see an overall signal for microglia. That doesn't imply that all the signals work through impact on microglial transcriptional regulation. And in this region here, which encodes two independent Alzheimer's risk signals, when you overlay it with either pseudo-bulk flow-sorted cell type-specific data or increasingly single nuclear ataxic data in this example, you see that actually these two signals are probably not acting through microglia. The one on the left probably through neurons, the one on the right probably through astrocytes. And then another use that we can play is as we develop cellular models of these systems that we can exploit in screens and so on, we can use those patterns of enrichment for the, from the human genetic data, in this case, for example, comparing Alzheimer's disease, GWAS, with various sources of microglia, both from primary tissue and from differentiated iPSCs, to ensure that those models are retaining the regulatory landscape that is most pertinent to those genetic signals. The second approach is then to try and go in and evaluate allelic function using single-cell genomics. Most obviously, you'll be familiar with the use of that for regulatory variants around single-cell EQTL generation. I won't talk too much about that. We're also using it in the context of a large consortium called the FinGen Consortium, which has built a large genome-wide association study in Finland, where we've identified now hundreds of largely coding variant signals that are associated with common diseases that we care about, and the big question of how to functionalize those at scale, which we're taking forward by recruiting carriers of those genotypes back in, and many of these are genotypes that are enriched in Finland because of the demographic bottleneck that the Finnish population went through. So this can really only be done in the Finnish population. And then by collecting biofluids that we can take out for proteomics and metabolomics and the rest, and cells that we can take forward for single-cell transcriptomics and cell painting and the like, we can start to functionalize these variants at scale and understand exactly the mechanisms through which they're impacting disease risk. Now, as you'll appreciate, if it takes millions of samples or hundreds of thousands of the samples for us to find these genetic signals, they're often pretty weak at the whole body level, and some of their molecular effects will also be weak. So there's a lot of merit in combining these together. You'll be familiar with the idea of polygenic risk scores, where we can take an individual and score them for the sets of variants that they have that predispose to disease, and therefore get people on the spectrum from those with a high level of genetic predisposition for those to the low. And we can then exploit the power that comes from aggregating these variants, which are individually of quite small effect, by using village in a dish pool differentiation experiments, which are now becoming feasible, to explore the effects not only of individual variants, but these collections of variants on differentiation and other cellular phenotypes. And that's, again, something that we're exploiting at the moment in relation to both age-related macular degeneration and Alzheimer's disease. Now, even when you have really good evidence of a genetic signal that co-localizes, for example, with a single-cell EQTL, that on of its own doesn't necessarily give you the confidence in the causal relationship. It could, of course, be that that variant is influencing the single-cell EQTL expression in a relevant cell type and influencing disease risk, but they're not on the causal pathway. There's a lot of molecular pleiotropy out there. So ideally, you're going to want to confirm those inferences through some sort of perturbation study. Sometimes nature's done that experiment for you through coding variation that you can detect through human genetics, but otherwise you'll need recourse to experimental perturbation of genes and the cis-regulatory elements. And again, increasingly we do that using single-cell approaches, and the overall plan is the one that's shown on the right there. Take a GWAS, find the signals, fine map them so you get down to the level of the variant, and then come up with inferences based on the overlay with single-cell atlas data of various kinds of the cis-regulatory element that's involved, the likely effector gene, and the cell type and state in which this is active, and then seek to confirm those by perturbation experiments of both the gene level and the enhancer level across a range of cellular models. Central to that, of course, is to have good central cellular models, single cell, co-culture, organoid, that recapitulate as many of the features of disease biology as you hope. We're increasingly able to do that, in this case for microglia, and then undertake large-scale perturbation screens as shown here. And in terms of an overall framework of really what we're trying to do, we have these genome-wide association screens for both common and rare variants. We can see those as in vivo human screens of natural genetic variation for clinically relevant phenotypes. We're increasingly able to take that inventory of genetic signals and partition it based on cell type specificity, pathway membership, overlap with other phenotypic features into different partitions of risk which capture different processes that might be involved in disease risk. And we can use those partitions and the risk scores that come with those partitions as a coordinated metric for evaluating the consequences of common natural variation, whether that's at the level of whole body individuals where we can capture clinical and proteomic phenotypes and the like, or of cells or organoids derived from those individuals where we may be able to capture more molecular and cellular phenotypes. And then we can anchor all of that with large effect perturbations from both chemistry and from engineered variation, which can give us some of the inference to lay on top of that. The goal here, of course, is to get to a multidimensional description of that connects perturbations in phenotypes to causal, molecular and pathophysiological processes. So in the last couple of minutes, let me just tell you one story that doesn't really have a genetic basis, and I'm indebted to Graham Heimberg, who's in the audience, who was one of the main protagonists in the work, and I'm sure he'll be able to answer more detailed questions on this than I. So there's a lot of interest in this population of fibrotic macrophages that's been seen in many studies, including some from the HCA groups. It's seen to be enriched in a number of fibrotic conditions, in a condition called idiopathic pulmonary fibrosis and another called pulmonary or systemic sclerosis. And begs the question as to whether those macrophages are causal for disease in some way, whether they arise as a result of disease and maybe represent some sort of protective mechanism, or whether they're just innocent bystanders with no causal connection to disease pathogenesis. And the genetics here so far hasn't been particularly helpful. So one of the ways we might try and understand a little bit more about these cells and what they're doing and answer those questions to do with causality is to understand where else we might find those cells. And to do that, we need two things. We need a big collection of diverse single-cell RNA-seq and other genomic, single-cell genomic data sets. And we need a similarity metric for single-cell gene expression profiles. So we can ask questions about where else have we seen similar cells. So this is the work that Graham's been doing since he came to Genentech over the last few months. Firstly, scraping a lot of public data sets to get access to large amounts of single-cell expression data, 89 million cells so far and counting, parsing that, harmonizing the data and the metadata to generate human tissue-specific single-cell atlases. And then in terms of the similarity metric, borrowing ideas from the Google reverse image, where a query image can be interrogated against the vast repository of other images using deep learning methods. And that has some parallels with what we're trying to do here. Can we take a query single cell, can we embed that cell into a common latent space and use it to find similar cells? And the trick that's used there, which was used in the Google image, is to form these triples, to find pairs of similar images, and then use deep learning methods to identify the features which are similar between similar images and distinct from disparate images. And that's the approach that was taken, using cell ontology as the way of identifying the similar and dissimilar images to train a model. And this is that model here, in this case generated with 13 million cells from 340 studies, now all projected into one common space. So with those data, you can start to ask questions, where in that common space are these fibrotic macrophage-like cells? And they map in that circle there. And you can ask, well, what studies generated those cells and contributed to that part of the plot? And it turns out these fibrotic macrophages are present in hundreds of different samples from hundreds of different studies. So then you can ask questions, well, where do we see particularly large enrichment of those fibrotic macrophages. And you can see in the top there, they're particularly enriched in lung, and within lung samples, they're particularly enriched in fibrotic lung. Well, that makes sense. That's not a big surprise. They're also enriched in COVID lung studies. They're also enriched in certain cancers, such as in this pancreatic ductal adenocarcinoma. And this gives you some clues then as to what these cells might be doing in a number of different disease conditions. And then finally, wouldn't it be great to have a cellular model of these cells that you could use for mechanistic inference? Turns out that when you look at those studies that are contributing to that part of the plot, it includes a number of in vitro studies that inadvertently also generated FMP-like cells, and we've been able to use that to generate an internal model now by recapitulating those protocols that inadvertently led to these cells in those other studies, and use those for further mechanistic inference. So let me finish there, recognize all the folks who've contributed to the work, highlight that I think there's great opportunities as we bring these worlds of human genetics and single-cell genomics together, and then highlight some of the areas where we need to continue to develop to flesh out this work. Thanks.
Mark, thank you very much. Are there questions from the audience? Or I have one, and then we have I'm always putting this up when it comes to genetics. What about environment?
Environment's important, yeah. And if I'd had time, I would have given you a whole spiel about how genetics is everything, but
got it.
There wasn't time.
Please.
Hi, I'm Mike Fletcher from Nature Genetics. As I'm sure you know, biobanks contain a lot of other rich phenotyping data at above the cellular molecular level. Could you perhaps, I was wondering, how are you analyzing that data in the context of, especially in the context of the HCA, where we've got these, trying to get across multiple scales from tissues all the way down to the molecules, how are you approaching that?
Well, I did have a slide, the slide I had halfway through, trying to think about how do we put these things together, and certainly one of the areas that, from my previous life in diabetes, where we were able to partition all of those genetic signals for type 2 diabetes into the different processes and different cell types through which they were mediating. I think that's a very useful way of thinking about it, that we can strip out that genetic risk into the different components. And that then, as we bring in the cellular and organoid data, we can overlay that. And then we're not just simply looking at the relationship between overall polygenic risk and, for example, beta cell phenotypes in the case of type 2 diabetes, which will give you a lot of noise because a lot of that genetic signal is not coming from beta cells, it's coming from fat and muscle and liver. So I think it's that decomposition that can be pretty powerful at cleaning up the signal and therefore allowing us to align the clinical data with the molecular data in a far more tangible and effective way.
Dana.
So, beautiful work on really, if anyone didn't notice, I think querying is important. So where is my cell? Great job on that. But one of the things I might have missed, you could identify your cell in a very large data set very effectively. But what did the model itself teach you biologically about the cell beyond being able to sort of really characterize where it is exactly?
Bit too early to say. That was hot off the press. I mean, this data was generated pretty recently, and those data that I showed you very briefly, showing that we'll be able to recapitulate the differentiation and produce these FMPs is generally in a few weeks old. So I don't have anything to tell you yet, but it's really great to, unless Aviv knows something I don't know, which is very possible.
But I actually comment on that just from looking at the slide, right? That these are macrophages that people mostly thought about in the context of lung fibrosis. Now, all of a sudden, you see, in fact, they also exist in pancreatic cancer, which is actually a very fibrotic cancer. Yeah. So all of a sudden, you're like, maybe it's lung fibrosis, maybe it's just the fibrosis which matters to them. So even in the simple, without looking back at the model, just using the query to find your cells, you at least learn more about the characteristic of those cells beyond the original place you found them, and it expands your context.
That I noticed, and that was beautiful. I guess, again, I'm putting you on the spot, and this is new, and maybe we can put Graham on the spot.
Yeah.
how do we get to the genes that really, and the processes inside the cell that really define the cell? So now, we know this is a fibrotic cell. These are all the different contexts and places that you see it, and that's very valuable. I tried to start, by saying that, but then, how do you connect that back to the genes, the gene programs, and what internally defines this cell type from, from the model itself? And anyone involved.
Yeah, well, most obviously, of course, we can do screens now to see whether we can influence the rate of differentiation to that phenotype, and also we can do screens to see to what extent we can remediate that phenotype. So that's the obvious things that are going on.
Mark will bring that next time.
Yeah.
Thanks again, Mark. The last talk is online from Eli van Allen from the Dana-Farber, and he will have a pre-recorded talk on leveraging single-cell technologies to understand clinical resistance to cancer therapies in urinary cancers. Start.
Thank you for giving me this opportunity to present. My name is Eli Van Allen, and I'm a medical oncologist at Dana-Farber, and I run a computational biology and cancer genomics lab, both at the Farber and at the Broad. And I have an interest in understanding clinical resistance to therapeutics in cancer generally and in genitourinary cancers in particular. And it's a privilege to be able to present here today with this group to talk about some of our work using single-cell approaches to understand this major challenge in the field. There's my lab website if you want to check it out. And of course, my Twitter account where I periodically complain about our electronic health record. First, here are my disclosures. So in terms of intrinsic and acquired resistance to cancer therapies, unfortunately, just to sort of set the stage for this audience, in medical oncology, when we're met with an advanced prostate cancer patient or advanced cancer patient in general, we have to consider a series of options, and we have actually a whole effort in our lab to try to pick the right option amongst a series based off of molecular and clinical features. But we know that even when we do this, more often than not, the patient will become resistant to whatever we give it. And that resistance can come quickly, that resistance can take a long period of time. Every once in a while, we'll meet a patient in whom we give a therapeutic, and this patient has an exceptional or extraordinary response that's durable. And we want to understand all these different manifestations of clinical resistance to cancer therapies in patient-centered ways whenever possible. And so one of the open questions that we've been trying to think about over the last few years is whether single-cell approaches can help us understand clinical resistance to every therapeutic modality that we give in the clinic, targeted therapies, chemotherapies, immunotherapies, combinations therein, and how we can actually use this information for biological discovery and perhaps clinical translation. And nowhere is this more evident, and certainly amongst many places, but this is a place near and dear to my heart, than in prostate cancer. And just to give a quick primer as to how we think about advanced prostate cancer and metastatic progression, it's commonly the case that in metastatic prostate cancer, the lesions will move from the prostate to lymph node, bone, liver, and rarely to other organ sites. There's a diverse series of molecular events that have been described after patients become resistant to the therapies we give them, when they become so-called castration-resistant prostate cancer. And in some cases, these tumors will actually demonstrate lineage plasticity and switch to a neuroendocrine cell type that is extremely aggressive and highly lethal and typically refractory to all therapeutics that we give it. And this is outlined in this cartoon schematic below. And despite numerous advances over the last decade to advance the therapeutic compendium in this disease, we still have a long way to go. And in most cases, castration-resistant prostate cancer is lethal. And so with this as a context, we embarked upon a study quite a while ago, actually, is when we started it, as evidenced by the single-cell sequencing modality that we chose, to basically understand the single-cell landscape of metastatic prostate cancer, and more particularly, to understand transcriptional programs that might be convergent on resistance that we can actually use to come up with new therapeutic paradigms for this disease. And so we had biopsied about 15 or so patients with metastatic prostate cancer pre or post therapeutics that we give in the advanced setting. This is led by Dr. Meng-Xiao He, pictured below, along with collaborations involving Dr. Aviv Regev and her team, Dr. Mary Ellen Taplin and others at Dana-Farber, and many others. And here's our sort of summary, just representation of the different cell types that we observed in this setting. And what I want to zoom in on is actually the tumor cells that we're trying to study in this case, because that's oftentimes where a lot of the therapeutics advances have come from in this specific disease. And the predominant therapy that we give is hormone-based therapy that targets androgen and the androgen receptor in particular. And so where we started in looking at this data set was looking at different modes of androgen receptor splicing, alternative splicing isoforms that have been described, and whether those actually tracked with different resistance patterns clinically. And so you'll see on this plot, the sort of organized, each sort of suite of columns is really about a different patient and a patient biopsy that's split between exposure or non-exposure to these different therapeutics. And the details, in the context of this talk are not relevant, but the aggregate finding that in essence, the splicing patterns were diffuse and sort of not really segregating between pre or post enzalutamide, which is an androgen-targeted therapy, really told us that if we're going to maximize our insights from this data set, we're going to have to zoom out and go beyond sort of androgen receptor biology to make sense of what was really happening in the transcriptional space in this disease. And so what we did was we actually took the full set of tumor cell data that we had and developed an approach to try to overcome some of the patient-specific confounding effects that can arise when trying to do comparisons between groups, and came up with a statistical strategy which is described in some detail in this Nature Medicine paper, but also in a preprint that's listed below. In essence, where we came up with a subsampling strategy to overcome this issue. And we projected into this a set of hallmark cancer gene sets and some curated sets that have been described as possibly being relevant in prostate cancer. And here's our aggregate results, which obviously don't have time to go into detail, but I want to pull out one finding from among these that was actually surprising but interesting, which was specifically a TGF-beta signaling program being operant as elevated in the post-enzalutamide castration-resistant setting relative to pretreatment cells. This has actually been observed in immune cell populations in mouse model systems of prostate cancer, but to our knowledge had not been described in tumor cell intrinsic ways in patient biopsies in this particular setting. And so this is what it looked like in a little bit more detail. Again, not a profound difference, but seemed to be some sort of a shift between the pre and post-exposure cells that we actually could validate in some cases with immunohistochemistry, and perhaps more provocatively in some cell line systems where we not only evaluated the signaling effect of this specific program in a few different prostate cancer cell lines, we also explored the use of therapeutic inhibition using TGF-beta selective inhibitors in this context, which we think might have promise should there be more focused and sort of less toxic affected TGF-beta inhibition strategies in the clinic moving forward. In addition to looking at tumor cell intrinsic biology for sort of shifts in programs in the adenocarcinoma space, we also wanted to look, because we had these data, to try to get a sense of some of the lineage plasticity properties between adenocarcinoma and neuroendocrine prostate cancer, because we had this in our data set and we could actually ask this question. And so what we tried to do is go from individual genes into regulatory networks and make sense of this both from the single cell data and also project this into a bulk transcriptome sequencing data set of adenocarcinomas and neuroendocrine prostate cancers that's on the y-axis here. And so, for instance, regulons that are down in small cell prostate cancer involve pretty canonical androgen receptor signaling genes like ERG, ETV1, and so on and so forth. And then the ones that were up in small cell on the top right of this plot includes some genes that are actually pretty well described in this setting, EZH2 and SOX2 in particular. But the strongest ones were actually genes that were kind of surprising to us. HOXP5, HOXP6, NR1D2, which actually has a therapeutic that's being tested in non-prostate cancer settings. And wanted to dig further into this, and so we actually collaborated with Misha Beltran and her team. We're able to do some confirmatory staining in both adenocarcinoma cells, but also in neuroendocrine prostate cancer-derived organoid cells to really confirm the presence and relevance of these particular regulons in this context, and are now doing further biological investigations to make sense of this, but use this more as an illustration of how we can use patient-centered single-cell analysis to make biological insights that can lead us into novel and exciting directions. We did not have much representation of immune cells, generally speaking, in this data set, but we obviously wanted to look. And indeed, so one other interesting finding we had in this context was looking in our T cell compartment. There was a subset of tumor cells where we actually had pre and post enzalutamide exposed cells from the same patient. And we could describe evidence for T cell clonal expansion in a subset of these tumors, which was quite surprising to us that we might be able to see this kind of an effect, which we could actually validate in a subset of bulk RNA sequencing data sets from other cancer patients. This is something we're trying to wrap our head around in the context of a relatively limited clinical impact of immune checkpoint blockade in this disease, but perhaps suggesting that there may be some relevance to looking at therapeutic strategies in series rather than in parallel, which is how we oftentimes think about combinations in oncology drug development, because in some cases, one drug may be shaping the microenvironment of a tumor that may actually have impact for the relative benefit of the second drug, but only if it's done in a certain order. That is speculative and still sort of something we're all trying to make sense of, but again, it was the kind of thing that we could draw insights from using these single cell data. This is just one quick vignette. Obviously, you don't have time to go into lots of other things that we're doing in this space that we're really excited about. This is actually part of an effort that we're doing across Dana-Farber and the Broad through the Center for Cancer Genomics, whereby we're trying to get biopsies pre, on, and post therapies in metastatic settings across a number of different contexts. Historically, of course, doing bulk sequencing, but increasingly doing single cell and spatial analysis paired with model systems. Another example, which I don't have time to go into in the genitourinary cancer space, is looking at immunotherapy resistance pre and post therapy, for which the citation is listed below, also from last year, led by Meng Xiao and Kevin B. All of this is really under a paradigm of expecting patients to come to us to participate in this type of research. But of course, we recognize this exacerbates disparities in the kind of science that we generate. And so what we're also trying to do is come up with a way to actually engage with patients and meet them where they're at, rather than have them come to us to participate in single cell projects and so on and so forth at Farber and other select institutions. Can we actually bring the research project to them? And so what I wanted to end with was just describe very briefly the Metastatic Prostate Cancer Project, or mpcproject.org, which is part of our Count Me In initiative. And in this case, a patient, wherever they are, currently in the United States or Canada, but hopefully as we expand more broadly, can go to this website, click the Count Me In button. We then ask them to fill out a simple online consent form, and we ask them a few questions. We could then start to send them things, saliva kits, but also liquid biopsy kits that we can use to generate all sorts of molecular data, bulk, cell-free DNA, single-cell analysis of circulating samples, along with getting a permission to access their archival tumors when available, and use this to drive molecular insights. And we've had over 1,100 men sign up so far. And actually, here's the full map as we have it, which is pretty exciting because it's evident, even just visually, that we're capturing a different swath of patients than we tend to study in these types of research programs already. But we also have more data to more clearly delineate this. And so, for instance, in our first freeze of data, we noticed that the vast majority of our patients are cared for at non-NCI designated cancer centers, and a reasonably large minority are from either physician shortage areas, medically underserved, or rural patient populations that, again, tend to be the ones that are less represented herein. We have a long way to go, and in prostate cancer is emblematic of a context where there are massive disparities based off of race and ancestry sort of impacting this in ways we don't understand, and so we have a lot of work to do. But we're excited about this as a paradigm to help us overcome some of these disparities going forward in this type of research. And on that point, so here's just an end of one example of a patient who received a swath of different therapies, including sort of an experimental TGF-beta inhibitor along with immunotherapies. This is a prostate cancer patient where we could actually use some of the molecular data to derive clonal inference about tumor phylogenetics over time, ideally using single-cell analysis going forward, and that's really where we'd like to move in this space. So with that, again, thank you for giving me this chance to present. I hope one day to see you all in person, hope all are as well as can be expected in these constantly uncertain times, and happy to follow up any point in time. There's, again, lab website, Twitter handle, and again, thank you so much for this invitation to present some of our work.
Eli is not online, as I understand. I think if he's in Boston, it's 3 o'clock in the morning. There will be no questions, but he said to us that he would like to answer questions. You have them, just send him an email, and he will be happy to answer. That's the end of the first session. I think we got 3 exciting examples how single cell will enter the medical arena. And there will be more, and there are more outside. We have now 30 minutes coffee break, and then you see on the slides here for the breakout session at 10:35 where you have to go, and we'll see you there again.